Yeah, Katja, thanks a lot for this very kind introduction and for the invitation. And first of all, congratulations to the Institute here. It looks really nice. Katja gave me a tour. It's great. So today I would like to discuss with you an idea, a new way, hopefully, to neuromodulate in a non-invasive way, in a relatively focal way, the brain structures. And how we can hopefully modulate human behavior with this, and I provide you there some first approaches in humans to go this way. I guess. Oops. Sorry for this. Okay. Okay. So yeah, in everyday life, it's very important to learn and to memorize new information, and this is from young age to old age, and it's also quite important in terms of neurological disorders. So, for example, as Katja said, I worked for a long time in the stroke recovery field, and their learning processes, or if you want to call it relearning processes of lost skills, are a core part of the rehabilitation process and of the recovery process. So in this view, it's important to, on one hand, understand processes underlying these learning and memory processes. And here are experts, a lot of experts in this field. But also one idea is how we can hopefully modulate these processes to improve behavior or to improve recovery from lost functions due to a brain lesion, such as a stroke or a traumatic brain injury. One important point in this direction is clearly that the brain works as a network with different areas working together, orchestrate their interactions to best implement the function, for example, for the upper extremity. And here is just one kind of display of the network. You might see there is like you have bilateral primary and secondary motor areas active, but also subcortical areas, cerebellum, which you don't see here, but is also involved in the implementation of such tasks. So one question is if we can neuromodulate part of structures of this network. On one hand, can we improve behavior, but also on the other hand, if it gives us additional information of causality of such brain regions in the network and how they impact on parts of this process of acquiring a skill or encode a skill or memorize other functions. And for a long time, I would say the main technologies used to this are basically transcranial magnetic and transcranial electric stimulation. I don't want to go too much into detail. I guess most of you are probably familiar with this. Maybe most used is direct current stimulation. So you basically apply either via two larger electrodes or a set of electrodes to have a more focal field, a direct current in the range of a few milliamps. And depending on the polarity, you can up or down regulate basically cortical excitability for a certain amount of time. Or an alternative technique is transcranial alternating current stimulation. Obviously, you apply alternating currents with an idea of probably entrain ongoing brain activity. And then we have transcranial magnetic stimulation. Also, in terms of neuromodulations, there are different types of protocol, depending what you want to achieve from simple repetitive stimulation, either low frequency or high frequency, which kind of results in an inhibitory or excitatory effect, or more patterned applications like a theta burst application, where you have, for example, a high frequency burst arranged in theta patterns. And then depending if you have these patterns in a continuous way, it rather leads to an inhibitory effect, or you apply it in an intermittent way rather to an excitatory effect. And this concept of theta burst is basically based on this very classical LTP induction protocols, and I will come later on to this back in a minute. So I want to just give you very few examples how these techniques then have been used or can be used, for example, to modulate motor skill acquisition, motor learning, and it's mainly then applied to either the primary motor cortex, contralateral, usually contralateral to the training hand, if you want, for upper extremity, or to the cerebellum. I will give you also another example where an impact more on frontoparietal interactions in terms of learning, with a focus on whether you train a sequence based on a memorized sequence or very explicitly presented sequence. So in this regard, in the last years, we looked in a bit more detail in these learning processes of an explicit sequence, like a piano sequence, in different age groups. And what you there can clearly see, if you train the patients for around, or subjects for around half an hour per day, you see that they improve, all of them, even the healthy older, right? But the dynamics of the improvement is fundamentally, or it's quite different between the groups, especially if you focus on the more extreme two groups, like the young ones, which means between 18 and 30 years old, or the older group above 60 years up to 90. And what is very clear is that especially this very early on phase, the first day, this fast online learning is quite different from young and older subjects. So the young improve quite a lot there, and the older ones few, and they kind of improve over the 5 training days in a more constant way. And the second striking point is that the overnight consolidation is clearly known and apparent in healthy young subjects, but it's absent in healthy old. So one question which came up, and there were first smaller studies with shorter trainings, if we kind of stimulate the primary motor cortex in combination with such a skill acquisition task in healthy young or healthy older, especially we were more interested in healthy older, whether we can improve basically the acquisition of such a skill. And as you see here, again, this is the same kind of task. The subjects performing either with a placebo stimulation, and you see the same kind of improvement like in the previous study. However, if the training is combined with a nodal transcranial direct current stimulation of the motor cortex, then you see a clearly different kind of improvement over time, with especially a large change in the first day of training. And if we call back the subjects 10 or 60 days after the training, this effect is still there. So it doesn't decay over the time back to the level what the training was with the placebo condition. What was quite interesting is, or what we saw in healthy young subjects, if they trained this first day, that in this first day, they very fast improved their accuracy in this type of task, and the speed continuously was improved over training days. In healthy older, in this condition without stimulation, what we saw there is that both of these improvements were more in a slower, not as steep way. Combining stimulation in the healthy older, combining in the healthy older stimulation with the training, then we found a quite different pattern. What you can see here in the full lines. So the accuracy, quite comparable to the healthy young, improved then in this early phase of learning, quite steep. And here you see the curve for the placebo stimulation. And the speed continuously changed. So overall, it appeared from the stimulation that it rather impacted on the accuracy, and the speed continuously improved than other effects there. So one question appears also, and I want to raise this only very briefly. If somebody's interested, we can discuss it later on a bit more in detail. Is really age per se the problem, let's say, or the factor which impacts whether stimulation leads to an improvement or not, or other maybe the baseline performance. And we took basically the whole group of subjects and looked at baseline for parameters of performance in terms of accuracy and speed. And we could then separate by a clustering process basically 4 groups, depending on how their baseline performance is. And then you had this optimal performance early on. And these subjects, they were mainly young, but there were also older ones in there. They didn't improve from the stimulation at all. It was rather disturbing. And then if you take the other extreme of subjects, these were the ones at baseline who didn't perform very well in terms of these parameters we chose. And these were the ones who profited the most from stimulation. So probably age per se is maybe not the core factor, but basically the baseline performance of the patients, of the subjects for such a task. Okay, then I just want to give you one brief other example. So it's in a way a comparable task like here. The only difference is that in one part of the task, like here, the subjects had to memorize beforehand the sequence they have to play, and they played it basically from memory. And here in the other condition, there were basically no memory load. And the hypothesis was that the frontoparietal interaction for working memory plays a role in this memorized condition, but not in the non-memorized condition. And this is again a cohort of healthy older subjects. And as has been shown that there are theta interactions between frontoparietal areas and are relevant for working memory. So we let these two groups basically train the task either during sham stimulation or theta transcranial alternating current stimulation bifocal frontoparietal in an in-phase fashion. As you can see here, as predicted for no memory load, the stimulation didn't play any role. However, in the condition where memory load played a role, subjects who received the varium stimulation performed the task better over the short training phase. But just to show you the different approaches one can use in terms of the cortical network partners in the motor network. However, I mean, obviously, deeper structure, especially the striatum, has been shown in a lot of animal and human work, plays also a core role in the implementation of such a motor skill, or in the acquisition of a motor skill, in the memorization of the motor skill. So it would be obviously an interesting target for neuromodulation, and see how much it then impacts directly in humans on behavior. And it would open up a new way to address also the whole network, and probably probe in a more causal way the functional role of the striatum in humans. So can we do it? So one way so far how it can be done is basically by deep brain stimulation in an invasive way. Obviously, this is limited in a way to patients who anyways need this type of stimulation, like Parkinson's patients, right? And it's obviously not a way to go for healthy subjects. So the question is, do the current brain stimulation techniques offers also the possibility to target this deep structure? Well, simple answer would be yes, it can be targeted, but it has a high price. And the high price is basically non-focality. So there's a very steep depth-focality trade-off for electric or magnetic stimulation. So if you target basically deep in the brain, then you stimulate the overlying cortex in a very large scale. Even with, let's say, more advanced coils like H coils, etc., structures to reach structures like the striatum in a relatively focal way is probably not really possible at this stage. So it's a clear disadvantage, and it does not allow to use these techniques in a kind of focal way to modulate structures like the thalamus, like the striatum, etc. So, but I think there are probably two new methods which hopefully in future will allow this in a bit more detail, also in humans. And so one way is obviously focused ultrasound. For neuromodulation, I mean, ultrasound to do brain imaging in clinical neurology there since long time. For lesioning, also quite long there. Now, blood-brain barrier opening is a new target, and neuromodulation. I think it's quite interesting in a sense because you can be very focal with an ultrasound beam. I think a bit still the disadvantage, I would say, is that the mechanistic understanding is well, let's say limited still. There are several hypotheses how the neuromodulatory effect is implemented by focused ultrasound, and I think the parameter space is also quite unclear. And if you think in terms of that you would like probably to apply a neuromodulation part which is physiology inspired, so more like mimicking or very close to brain processing, I think this is also not so easy to achieve with focused ultrasound. There are first attempts in the motor cortex one. I think Robert Chen showed nicely that in a type of a theta burst mimicking focused ultrasound neuromodulation, we could show to modulate the motor cortex. But I guess it's very interesting, but there's still a bit of basic work needed to be able then to apply it in a larger scale possible. I will not discuss more about this. If you're interested in more discussions, we can do this later. Okay, then a few years ago, from Neil Grossman, he came up with a quite new idea of how to apply electric oscillating currents to the brain and arrange them in a way to reach deep structures, so-called temporal interference stimulation. They showed nicely in a rodent model that was able to And I'll show you in a second the basic idea behind it. But they were able to show with this technique to reach deeper structures in rodents and have it in a fairly focal way, and that they could steer basically the field deep in different structures to modulate behavior. So then the question is, can we easily, or is it possible to translate this also in humans? And I want to show you first attempts here now. So the general concept is based that you apply high frequent oscillated currents. So high frequency means in the kilohertz range. And due to the filtering properties of neurons, usually in this high frequency neurons do not respond to this oscillating electric currents. So that's the first point. So that you can apply, for example, through two pairs of electrodes, these high currents, and then at some structure, depending how you arrange your electrodes, these two currents, these two oscillating high frequency currents, they basically interfere. And so now, if you have two high frequency currents, which have a small difference in frequency, and then you overlay them, so then you generate basically a beating envelope in this small difference between the electrodes. So for example, if your carrier frequency is 1000 hertz of one electrode pair and the other electrode pair 1010 hertz, then you get a beating envelope in this difference of the frequencies, which then is in a range which probably neurons respond to, let's say 10 hertz. So in principle, something like what you can see here, right? There's a small difference in the frequency, and then you see basically this beating envelope. And this is what they basically promoted and showed in this previous work. So for example, then depending on your arrangement of your electrodes, so you can basically have this interfering field at a certain structure deep in the brain, and you have there this beating envelope there. While you have in cortical areas these high frequency oscillations, which are supposed to not activate neuronal structures. So just to give you a bit insight from their original work, what they did. So what probably is interesting also, depending of basically the ratio of your currents, you can steer basically your field of the beating envelope to one side or the other, depending if you change here, what they can nicely show. And then induce the respective movements. Was quite interesting in a very new study from Ines Violante. They did basically a cadaver experiment and compared classical transcranial alternating currents with a certain frequency versus temporal interference stimulation, which is supposed to achieve deep in the brain the same frequency, and calculated there the electric fields. And the amplitudes, and as you can nicely see here, or if you take classical AC stimulation, you reach the deep structure. In this case was the hippocampus. However, in the cortex, you have even higher intensities. In contrast to this, and this is the goal of the transcranial temporal interference stimulation, that you reach this deep structure with high enough magnitude, but without co-stimulating the overlying cortex. So this was another step in the direction, say, well, probably this is an idea which might work out also in humans. Okay, so I guess from this first, let's say, evidence, one could say that there's a fairly good spatial resolution even to reach these deep brain structures. I think what is for humans very critical is that you can do reasonable simulations, how to place your electrodes, to reach then, or to calculate your interfering field, that you get to the structures where you want to go. And there are possibilities. So for example, from Zurich, from Esra Neufeld's group, they have a high resolution model of a brain, and there you can, we did with them together the simulations, and you can simulation in one high resolution brain, and then calculate where you place the electrodes to target hippocampus or any other region. So overall, it looks quite promising. We can do the simulation. We have animal evidence. So now the question is it feasible to do it in humans, and is it probably also effective in humans? So just to give you a bit of a notion how these calculations can be done, the simulations, just one example here, or it's another one. It's a current planning in this platform where you can look for bilateral striatum or for the globus pallidus in a fairly focal way, I would say. Okay, so just to give you, I mean, that you have once seen basically the way to go. You need your two stimulators basically and the audio transformers, and then you bind them together, and then depending what type of stimulation you want, you program it, and then you can read it anyways out and to make sure that you get out what you want to have in an oscilloscope, and then you can arrange the way of stimulation you want, either whatever, a single frequency like theta frequency, or we used patterned stimulation. I will show you in a second what type of paradigm we chose and why. Okay, this so the following studies I want to present you the first attempts to apply this type of stimulation for neuromodulation of deep structures were spearheaded by three of my lab members. Max Wessel, he's now a postdoc, or he's continuing his neurological residency in Würzburg. Elena Peanato, she's finishing now her PhD, and Pierre Vassiliadis, who just finished his PhD. They were spearheading basically these projects, what you will see now in a second. Okay, so one important point definitely is to make sure that there's no placebo effect, and right, because if you want to have an interpretation of a behavioral result, it's important to make sure that everybody is blinded in a way. And for electric stimulation, what we know from DC, especially stimulation, but I guess also under certain, if you arrange it right on the AC, you can fairly well blind everybody. So I just want to show you the first results from around 120 sessions of TI stimulation versus high frequency stimulation. So in principle, you apply high frequency in, let's say, 1 kilohertz. And the only difference is that for the real temporal interference stimulation, you have 1 kilohertz and 1010, for example. So it's really a slight difference. And what you nicely can see here is that the perceived sensation, what the subjects kind of feel, they are dependent of the strength of the current, which is quite obvious, but there's no difference between the two core conditions. So here, this is a theta burst stimulation, TI, and this is the HF control stimulation. Or if you look basically of the reporting of the sensations during the sessions. And if you ask the subjects if they can distinguish between the conditions, that's not the case. So it's already a first good point to say, well, blinding most likely or perceived sensations of the subjects during a control condition and the barium condition is pretty well. Then just in terms of side effects and safety, there's a recent paper from Piao and colleagues, and they looked a bit more in detail on different parameters. So just for example, for the NSE, like a marker for a neuronal damage, where is it? Let's see. Oh, on the left, right? So it should be somewhere here. Yeah, okay, you spotted it. I didn't. It must be left. Here it is, right. Okay, for the NSE, there is no difference for the two stimulation conditions. And also, if you look on other kind of, let's say, missensations, is quite comparable between the active group and the sham group. So overall, probably the side effect profile is, let's say, like we would expect from oscillatory electric stimulation, and there's no hint so far, at least, that there's any damage to the neuronal tissue with all these markers we have at hand. Okay, so now the question for us was, what is a stimulation protocol probably which would fit in the ideas we are following up? So our idea was we were interested in the striatum as a core hub of this motor learning network, and ideally we would like to combine stimulation with motor training and see whether, on the one hand, whether we can modulate training, but also whether we see shifts in activity probably during this training process, or more or less activity in the striatum. So if you look on the in general, so one could think about like a more plasticity enhancing protocol which covers the whole training process. So in our case, it's around a half an hour of training or 20 minutes of training to do this. So to do this, we would like to have a protocol which is pro-plastic, probably like an LTP-like protocol. I mean, very classical, as you heard before, are the theta burst stimulation. So it has been established in hippocampal fields years ago, like 40 years ago, probably. And it has been translated, obviously, in the TMS scene, in the human non-invasive stimulation area. So with intermittent theta bursts, rather induce an LTP phenomenon or LTP-like phenomenon and a continuous theta burst rather than an inhibitory phenomenon. So this was the idea, and then combine it basically with the training paradigm I showed you before. There's also good animal evidence that LTP can be induced even in striatal region. There's a recent work from Hall and colleagues who showed that if applied theta burst stimulation, then even in striatal areas, they can induce LTP most likely and in NMDA dependent. So based on this data, on this idea, basically Max and Elena, they used this information and programmed our theta burst temporal interference stimulation in this way. So basically, you have as a carrier, we had 2 kilohertz, a difference of 100 hertz. So this is the high frequency part of the theta bursts, and arrange them that you have this theta trains either in an intermittent fashion, proplastic, or in a continuous fashion, which would be then rather inhibitory. And to test it, whether it works, basically we applied the whole paradigm during fMRI. So basically the subjects were in the scanner while they trained such a sequence, what I showed you at the beginning. And while they trained the sequence, we applied in the scanner theta bursts, temporal interference stimulation to the striatum, and compared it to a placebo control condition also during training, right? In principle. So the electrode placement was based on simulations, and then the subjects basically were in the scanner and trained several blocks of these sequences. Then in a second step for validation of the results, and for the goal, because we had the goal in mind also at some point to apply this, if successful, obviously in patients, was a second study where we performed basically the same experiment outside of the scanner, again with a training period which was even a bit shorter, applying then the theta burst and had post evaluations of the behavioral point. Yeah, just I can go quick over it. So in principle, this is the fMRI approach. The task is quite established. I think I don't need to waste much time here. So just the first research. So our very first question was: is the stimulation, or does it lead in the striatum rather specific effects? So for this type of task, one would expect that at this stage of the skin acquisition process, rather the putamen is relevant than the caudate, right? And now, if you look on basically the control condition, what you see in blue, can already see purely by the task without this additional stimulation, you see already a different activation of the caudate versus the putamen as expected. And then if you take into account the stimulation, then the stimulation effect was not globally on the striatum, basically leading to an enhanced activity in caudate and putamen, but it appeared it was only in the area and the putamen which was involved in the task. And we looked also in detail in subregions of the putamen, but there we didn't see any differences. So it was basically over the whole putamen. The second step, basically there were almost 15 years ago, a human study which showed in such skill acquisition process that you see a shift in activity within the striatum from the more superior, more associative, or my battery goes down, sorry.
Battery.
I'm sorry. From the more superior part to the more inferior sensory motor part. And so in our second analysis, we looked also if we see a kind of a shift in the striatum. So this was a very mild and it's a shift what we saw. So it's more a hint and it's a very hard.
Sorry. Yeah.
Yes, I have one.
Yeah.
Sorry. Okay, so it was a very slight shift, admittedly. So this is what we want to follow up in future direction, but at least it gave kind of a hint into this previously shown work from a French group in animal work. Okay, just to show you overall, then we looked of the whole brain activity and see that the motor task does what we're expecting, activating a bilateral kind of network with also cerebellar, etc. activity, as you can see here. If we contrast the activity during the stimulation, and again, this is task plus stimulation versus task plus placebo, then we see a few areas different. One of this is the contralateral basal ganglia striatum, but also like the SMA and some other areas, the cerebellum, which are within the motor network. So it appears that stimulation plus the task leads to not only an additional activation of the striatum, but probably of the network, of part of the network involved in such a task. Does the stimulation lead to an effect without the pre-activation of the respective network or the respective structure like the striatum? So basically, does it do the effect already during resting state? And to look on this in one way, sorry, I come to this probably now quick. So during resting state, we didn't see any activity. Admittedly, we did only a seat-based resting state analysis. There are other ways to look on it, but also based on some other animal work, which shows from Peter Fritsch, which clearly shows that for a lot of subthreshold neuromodulation, like this direct current stimulation, the pre-activation in principle is necessary to achieve the effect of stimulation. I think this research fits well into this area. So now the last step was we looked for behavior and association of behavior. First of all, there was a very small effect in behavior in the scanner, admittedly, in this healthy young subjects. However, the level of improvement with stimulation was correlated actually with the level of activity in the striatum. Okay, so overall, it was the first kind of promising result that we are probably able to target deep brain structures if combined with behavior. It has an effect on the electrophysiological or imaging parameters and a slight behavioral effect. So next question was to really validate this behavioral effect especially. And to do this, we were mainly interested in healthy older subjects because we saw from other studies for neuromodulation that effects of neuromodulations are much larger in let's say in systems, in situations where the motor system probably doesn't work totally perfect anymore. So in this study, we compared basically the stimulation outside of the scanner in a group of healthy young subjects, which a slightly smaller training protocol than in the scanner, was around 1/3 of the training from in the scanner. And we applied this also in a group of healthy elder subjects. As you see here in the healthy young, again, it was a shorter protocol. The effect before was already quite small, so we didn't see a clear effect. However, applying the same protocol for 7 blocks, it's like 20 minutes of trainings in healthy older subject. We saw the following. First of all, during the controlled stimulation, subjects improved also over time. However, the effect of the training was clearly enhanced with the theta burst stimulation, ATP supporting stimulation of the striatum in this group of healthy older subjects. So it kind of supported the idea that it's possible to target this deep brain structure and induce also behavioral effects. Now, open question is why for the healthy young, probably we didn't see an effect outside of the scanner. First of all, from an experience is that all the neuromodulation techniques we're using usually are in healthy young subjects have very, very, very moderate effects. And one explanation probably is that they train just much more in the scanner, like 3 times more. But this is also a kind of interpretation which had to be checked in future in more detail. Okay, so just a couple of potential influential factors. So again, perceived sensations in these studies were comparable. Then we looked also whether it impacts on attention or fatigue, right? I mean, there are some ideas that the striatum plays probably also a role in, especially in fatigue, but there's no difference for the stimulation conditions. And then we looked also for the focality of the field strength, either under the electrodes, or for example, comparing the SMA, this was one of the hub who was activated in the network, but the field strength, the simulated field strength is much lower than in our core area, the striatum, or if you compare amygdala versus striatum, etc. So was pointing that with the type of arrangement of the electrodes, we probably can achieve fields which are fairly focal in this terms. Okay, so now just to probably it's more like an outlook, because obviously we cannot answer these questions whether it's rather the direct or indirect pathway. But what we think from the data and from previous literature is that what is needed to achieve the effect is basically a task-induced pre-activation of the striatum that the stimulation probably develops, if you want, its effect. And there are two arguments for this. The one is like this difference between caudate and the striatum, and the second one that in resting state analysis we didn't see any changes, which doesn't exclude it obviously, because it's one way of addressing resting state. But I didn't show here. We did also an electrophysiological classical MEP analysis of the motor cortex. There we also don't see any difference between HF and real stimulation. So obviously HF is not kind of cortically activating something what we didn't cover. Okay, just let me kind of summarize this part. So I guess it's a very promising opportunity. There are a lot of open questions, obviously, but from this first study, I think we have some evidence that it's possible in humans to target structures like the striatum with behavioral improvement. Important is it has to be combined with a behavioral task to reactivate the structure, and then kind of steer also the effect of the stimulation to the structure. And I guess it's so far as we know, a quite safe and well tolerated stimulation, where we can also have a good blinding, which is important in terms of placebo effects. So then I want to, because we're quite excited by this first results, but obviously then we were thinking about another kind of type of concept, whether we can see it also in a different paradigm and in a different way of stimulation. And especially Pierre in the lab, he's interested in reinforcement. So he kind of raised the hypothesis that probably disruption of high frequency stimulation of the striatum might lead to an impact on the reinforcement part of learning, whilst low frequency stimulation, for example, in the 20 Hertz range, should not impact on the reinforcement, probably only on the motor part. I will give you a bit of hints why we came to this idea. So that the striatum is involved in reinforcement learning and motor learning. I think there's very strong animal work, but also associative information from imaging studies and or from patient studies, as you can see here, quickly summarized. And so then there is nice animal study which support this idea of that high gamma plays a functionally quite different role in terms of reinforcement learning versus beta oscillations, as you can see here, for example, in this work from Berke and colleagues. And so based on this, we developed, or Pierre developed, the idea that if we apply continuously an open loop striatal stimulation at 80 Hertz, this will perturb the reinforcement dependent regulation of high gamma oscillations and disrupt the benefits of reinforcement learning. Why? Because this high gamma information, which you can see in animal work, is highly time locked. So, and if you continuously apply basically an external gamma, it might interfere, and this was our idea. With the view is, if this is the case, then we have a certain causal link between the striatum and probably high frequency activation and reinforcement, the role for reinforcement. So the stimulation I can skip. So just to let you know, again, here in simulations, you see clearly that basically the fields are much stronger for the striatum than, for example, other areas which are involved in such a task or under the electrodes. Okay, so basically Pierre worked since a bit on a task where he basically let subjects, if you want, follow such a target with a gripper task, and then he provides either reinforcement in terms of whether they perform well or not. And then he looks basically on the outcome. I'll see if I can show you quick just the video briefly get them. So basically, it's the gripper task, and then it's the pre-training, and then so we read out how well subjects performed, and they train basically with reinforcement this task, and then there's a post-evaluation. So with classical, it's something like this, right? So we were interested rather in this post phase. So after the training, whether the reinforcement led to a better recall than later on this behavior. Okay, so kind of same setup if you want. We applied also this kind of task in the fMRI scanner, why they performed it with the different reinforcement conditions and the different stimulation conditions. In this case, we had the 20 Hertz rather motor oriented, the 80 Hertz rather reinforcement, and the placebo condition in there. So I think I can skip this in the time. So let me stop over this. So now, what you can see here in the sham condition is that after with reinforcement, the subjects performed better, as expected. Then what you can see here, overall, the performance with 20 Hertz stimulation was at a bit lower level. However, the reinforcement effect was still preserved, right? As you can see here. And then in the 80 Hertz condition, there was no reinforcement benefit anymore there from a behavioral level. So as a first hint into the direction of the hypothesis. So again, then in summary, so it appeared from the behavioral results, the 80 Hertz stimulation rather disrupts the reinforcement part, but not the learning in general. And the motor performance was rather disrupted in principle by both of the stimulations, as you can see here. So, and then the question was, so what can we see in the imaging? So overall, if we did a whole brain analysis, actually we did not see a reinforcement specific effect or a stimulation specific effect or an interaction. So we could not see. However, if we took the seed in the striatum specifically and associated basically data changes of the bone signal with the changes of behavior, then there was an association between the changes in behavior, basically getting worse, and the changing in the bone signal. So as one hint towards the relationship between the stimulation effect and the bone changes. Okay, so just for the adverse events, there were no adverse events, and you see here also this is basically what the subjects described, but this is, I would say, very classical, what you would expect also from alternating current stimulation. So there's no strong hint of a problem, and also the subjects were not able to distinguish between the variant stimulation and the placebo stimulation. So just as a short summary here, so from the study, it looks like that the 80 Hertz stimulation specifically impaired basically the reinforcement learning of this motor skill, and it was not the case for the 20 Hertz stimulation. So it's another hint that probably we can focally modulate striatal activity even in a very frequency-specific way. And then obviously there are also network effects I haven't shown you for the time reasons here, but and the safety profile and the blinding is quite efficient. So how much time do we have?
We have almost 15 minutes.
That's it.
So
Okay.
perhaps we can maybe 10 minutes.
No, okay, super. Perfect. Okay, super. So now I showed you mainly results from the neuromodulation of the striatal part of the striatum. Now I want to show you in the last minute some first ideas of other deep structure to further validate in a way the possibility of address deep brain structures and to probably modulate other other cognitive functions like memory functions. And I want to show you this for two kind of task cases. One is like a spatial memory, and the other one is more associative memory. I want to show you the first results there for discussion. So overall, I guess quite clear. So for spatial memory, obviously the hippocampus and the rhinal cortex are the core areas. They build a complex and they implement it, and this is very well validated by either the famous kind of lesion studies, right? Or if you want, also by either animal work or imaging work. And this kind of grid cell typical pattern plays a relevant role in this type of task or cognitive task, right? So for this project, we worked together with a group of Olaf Blanke at EPFL, and they developed recently a task in the VR. So you have basically this spatial navigation task in virtual reality where you have to retrieve some spots. And they implemented this task that it can be applied during fMRI. And Yoon Moon, he basically implemented here an analysis where they can look for grid cell-like activity. I will not go into detail. It's a very specific fMRI analysis. For details, you will see here, or we can discuss it later. But now I don't want to go into so much detail because it takes quite a bit of time to describe this. However, the study was performed in 30 young subjects, and we have the different blocks here and applied basically again theta burst stimulation. So in the sense of again, with the idea to have an excitatory or inhibitory theta burst stimulation in a control condition, and we targeted basically the right hippocampal entorhinal cortex, if you want, or complex. And so what we could find there is that if the subject performed this task while varium stimulation or facilitatory stimulation versus the kind of control condition, then they were able to be faster to reach the target. They are supposed to go. And what was interesting is that there was a kind of association with the bold changes for the two conditions here in regard of the behavioral improvement, what you could see here. So it was, in principle, it gave a hint that the stronger the hippocampal activity was during the ITBS, temporal interference stimulation, versus the CTBS, the better the subjects were able to retrieve the information and to basically go to the target they were supposed to go. So in very brief here, so this kind of hinted to the fact that in a specific arrangement of the electrodes, also unilaterally, the hippocampus entorhinal cortex complex can be reached by the technique and modulated. And this had an impact on basically navigation behavior, spatial memory, if you want. There was an association basically with the increase of hippocampal activity by stimulation and the behavioral improvement. So, and then the last part to show you was a very simple associative memory task. So basically where you learn the association between whatever picture and the name of the T, or between a face and the name, is kind of a classical face naming associative task. And this was performed in healthy older subjects. So they had to basically train this task while stimulation was on. And then later on, they had to retrieve basically this information and provide whether this name and the face basically fit together or not, and they learned it beforehand or not. So these are the results. So during the training phase, we didn't see much of a change. However, if the subjects came back later on, especially 24 hours later, then the ones who trained the face name association with the stimulation of the hippocampus, they were better able to retrieve basically the information than the ones who trained basically under the placebo condition. And again, here, if you look on, because attention might play a relevant role here. So again, here you don't see any differences in attention or fatigue whatsoever, or sleep between the two conditions, the varium condition and the control condition. And again, the perceived sensations, they were also similar for the two stimulation conditions. But then I want to show you just as the last slide, work currently from Neil Grossman's group and Ines Violante. So they used a comparable task, but they applied theta stimulation through the hippocampus, right? Theta stimulation is highly associated in the hippocampus with the implementation of such a task. They applied this in healthy young subjects, and they looked in more detail also about steering. If you steer basically the temporal interference to subparts of the hippocampus, like anterior, medial, posterior, and they saw there also then different effects. So it pointed clearly to the fact that in the right way, you can even steer basically your signal to subparts of such deep structures. And they found also slight but behavioral effects on this face name task in their data set. Okay, so we summarize this. So I think it's another hint that probably with this type of stimulation it's possible to reach also the hippocampus and the impact on other memory tasks, which in this case was a face naming task. It looked like that it was not necessarily impacted on the training, that the training was much better in one or the other simulation cases, but rather than encoded probably better the information and made it then easier to retrieve it later on, like 90 minutes or 24 hours later. Obviously, the mechanisms are still open. Somehow either it enhanced the pairing here and consolidated this pairing better, and then allowed then a better kind of re-encoding and bring it up later on. However, this is definitely our topics which are now addressed or will be addressed in upcoming studies. We have no clear information for this yet. Okay, let me summarize. So I think overall it's a really exciting new possibility to reach deep structures in a way without, stimulating the full overlying cortex. So we have now at least first hints in humans that we can reach structures like the striatum, the hippocampal entorhinal complex, or the hippocampus with a fairly good focality-depth trade-off in this first proof of concepts. I think from what we know, it's safe, looks like safe. The perceived sensations, side effects, I would say now we have around in our lab 180 sessions or so. This looks quite comparable to what you would expect from alternating current or even DC stimulation. So it appears not to be different, but obviously, we need to acquire much more experience with this. But it opens this new opportunity that we can look from a scientific point of view more into causal relationships, right? And not only associative with kind of imaging and behavior, but really modulate behavior, either impair or enhance behavior. But there are a lot of open questions, very clear. So, and just to list some of them, I mean, we need definitely a better understanding of the mechanisms of action of this type of stimulation, that probably an envelope modulation is not exactly the same like a classical oscillatory activity. So there are probably some differences in the underlying mechanisms. It can clearly be optimized, the stimulation, because so far this models for the simulation is usually not an individualized model. These are high resolution models, but not individualized to the individual subjects. And if you think about probably patient applications, then even more might matter that you can do a simulation on an individual brain and not on a kind of on a model brain, right? Another step for improving focality is you can use, instead of two pairs of the two electrodes, obviously can use much more electrodes to steer your field much better, but it needs also then better simulations, obviously. And there might be also off-site effects, so off-target effects. So it's important in simulation to see that you not only target your get well to your target, but you might have an interference at some other parts of it. And then interesting, but this is probably a bit more a longer part, is then to see for state-dependent closed loop application. What I showed you here was everything open loop, obviously. Okay, then thanks especially to the team. They did obviously the work clearly, like Max and Elena and Pierre, Bayern, and then also thanks to the great collaboration with Esther and his team, near Grossman and Olaf Blanke and all our clinical partners. Thanks a lot for your attention. And we have in our new institute, if somebody's interested, we just launched a new fellowship program. Just have a look, and if you're interested, apply there. Thanks a lot.